Okay. All right. Thank you everyone again for joining. Today we have Dr. Robert Stern. He's a professor of neurology, neurosurgery, and anatomy and neurobiology at Boston University School of Medicine. He is internationally recognized expert on the neurodegenerative disease, CTE, and is co-founder and director of clinical research for the BU CTE Center. The focus of his research is to develop methods of detecting and diagnosing CTE during life and to better understand the risk factors for later life neurological consequences of repetitive mild traumatic brain injuries in former football players and other contact sport athletes. So today we have Dr. Bob Stern with us. Dr. Stern, thank you so much again for doing this presentation today. If anyone has questions, please put them in the chat.
Thank you very much, Simona, and thanks for everyone who's attending live. And for those of you who are on vacation and coming back and seeing this on recording, welcome back. I am delighted to kick this series off for the year and hopefully won't completely be repetitive for those of you who have heard several of us speak about CTE, since this is such a critical part of our overall center. And we have really the world's best people right here at BU studying chronic traumatic encephalopathy. Let me just start with some disclosures and point to some of my key collaborators at BU. These are only the ones at BU. There are many, many more people involved with all of our work. And by collaborators, I really mean the people who've done all the work, the people who are the brains behind it. I've just been very fortunate to be involved with so many people and to have so many really terrific individuals moving the entire game forward with our understanding of CTE. So what is it? Just a little background for some of you who might be new to this. Chronic traumatic encephalopathy is the same thing as what we've heard in the past as dementia pugilistica. It is a neurodegenerative disease that is indeed very similar to Alzheimer's, but it's a unique disease. And it's associated with a history of repetitive head impacts, including the symptomatic concussions, as well as those little repetitive hits that we refer to as subconcussive blows or subconcussive trauma. And when I say associated, recently there was a paper that was published following a set of criteria for how do you determine if there's a causal relationship for environmental and toxic type of factors for outcomes. And that paper, led by Chris Nowinski with a wonderful group of international colleagues, showed using the Bradford Hill criteria that indeed repetitive head impacts do cause CTE. And what happens is at the time of those repetitive blows, there's this cascade of changes in the brain that is set in motion that leads to the accumulation of hyperphosphorylated tau. And that continues to spread after the head impacts stop. And so it starts this thing. It starts this neurodegenerative process. And then years or decades later, it continues and continues, leading to neurodegeneration and the symptoms and signs that we think are involved. Like Alzheimer's disease and any other neurodegenerative disease, CTE can really only be diagnosed through postmortem neuropathological examination. And the first time this was done was in a case series in 1973 with boxers. And Corsellis first described the neuropathology of that dementia pugilistica, or punch drunk at that point. But it really is our very own Ann McKee, who has examined more brains with CTE than any other neuropathologist in the world. And BU has the largest CTE brain bank, the VA, BU Concussion Legacy Foundation brain bank, anywhere. With over 1,200 brains. I think it's even more at this point. And there are amazing colleagues that work with Dr. McKee that have really been leading the charge. Back in 2013, Dr. McKee and a bunch of us published this paper describing the spectrum of neuropathology in CTE. Since then, there have been two NIH-sponsored workshops for defining the neuropathological criteria for CTE. And through those workshops, the group of experts have reached a consensus that CTE is indeed a neurodegenerative disease. It's a unique disease from other tauopathies, including Alzheimer's disease, or other normal parts of aging, such as part or late. It's only seen in people with a history of previous brain trauma, usually repetitive. The pathognomonic lesion, the lesion that is specific to CTE, is what you see here. This is stained for the phosphorylated tau aggregates. What you see are neurofibrillary tangles in neurons, with or without p-tau in astrocytes. And the p-tau is deposited around small blood vessels and in an irregular pattern at the depths of the cortical sulci here. And it seems to follow a very distinct type of spread and progression, unlike Alzheimer's disease that can start centrally, moving out laterally and to the superficial areas. The CTE starts in these cortical depths in the frontal and temporal and parietal cortices. And then it starts to spread deeper, and eventually almost the entire brain is involved. And these stages of CTE, of these early stages of 1 and 2, and then the later stages of more severe disease of 3 and 4 have been validated in a study by Michael Osko and colleagues. So that's the neuropathology. More and more continues to be learned about it. And those pictures of the neuropathology have indeed gained a lot of attention. Some of the famous individuals who died and whose brains have been examined and found to have CTE, that kind of stuff has really led to early media attention, public attention. And the speed at which this whole thing took off was really unbelievable. The world started thinking about, oh, you know, something might be going on when you hit your head over and over again, as what happens in American football. But would the science itself also be as quick? And that initially, none of us could have ever expected, because science usually does go at a tortoise's pace. But in fact, if you look at the PubMed findings, if you do a search for chronic traumatic encephalopathy, just since 1987, you can see this unbelievable growth in studies and papers published having to do with chronic traumatic encephalopathy. And contrary to what a lot of people think, around 83% of these publications are not by those of us at BU. This is research that's done around the globe. And so, in fact, I think I would say that the science is moving pretty darn quickly. And yet, we still have some important unanswered questions and some key knowledge gaps, like, is CTE common? Well, research now is showing that it is pretty common in people who have a whole lot of repetitive head impact exposure, such as professional football players, maybe even college players. But what are the specific risk factors and resilience factors? We still don't know the details, and that's really important. It's not that everyone who hits their head a bunch is going to develop this neurodegenerative disease. And so work has been ongoing, and much more has to be done to look at the specifics of head impact exposure variables. Is it the overall cumulative amount? Is it the overall intensity of those repetitive head impacts, as a paper by Jesse Mez that just came out pointed to? Is it the age of first exposure to repetitive head impacts, something that our group has been looking at for many years? Is it only football, or is it other sports that have repetitive blows? And in fact, there's been more and more study and publications of other sports and other activities, including military exposure to repetitive head impacts. Sadly, I would surmise that intimate partner violence may also be a risk. But what about the genetics? Now, there's been recent publications by Jesse Mez and others showing that ApoE4 does seem to increase risk for CTE severity. It's not just Alzheimer's disease that ApoE plays a role in. What about vascular risk factors? That also seems to play a role in the development of CTE and the severity of it. And what about the lifestyle variables? We have to learn so much more about things like sleep and diet and exercise. And then what, if any, are the real clinical features caused by CTE neuropathology? Believe it or not, we're not completely sure yet. Even though I'm going to go into that in depth, I can give you a little heads up. We're really not that sure. And then most importantly, how can we treat or prevent CTE? That's really the ultimate goal. And so the critical next step is to be able to diagnose CTE during life. So what can we do? Can we actually diagnose CTE during life right now? Well, the short answer is no. We're getting pretty darn good at diagnosing Alzheimer's disease and some other neurodegenerative diseases during life because of the very long history of understanding the clinical presentation, and then over the last 10 years or so, the development of objective biological tests, of biomarkers made for those other diseases. But CTE, we're kind of new to the party. The longer answer to that important question, though, is we are indeed getting closer. So what's the first step? What is the first step in our kind of goal of being able to diagnose CTE during life? Well, first and foremost, we have to describe the clinical features and course based on whatever knowledge we have. And it goes back almost 100 years that we can learn something from. When the first paper so many of you probably have now become familiar with, called Punch Drunk in JAMA, published in 1928 by Martland, where he described the long-term behavioral, emotional, motor consequences of professional boxers, using such wonderful scientific words as cuckoo and goofy and slug nutty. And then he said, finally, marked mental deterioration may set in, necessitating commitment to an asylum. Soon after, the term dementia pugilistica was first used, the dementia of boxers. And then the term chronic traumatic encephalopathy, CTE, was actually first used in 1940 by Bowman, who described a 28-year-old professional boxer who had started to experience cognitive, behavioral, mood symptoms He presented with short-term memory problems, depression, irritability, some childlike behavior, and paranoia, none of which improved over the course of 18 months. This clinical syndrome of CTE in professional boxers has continued to have first the term chronic traumatic encephalopathy in the 40s and 50s, and then, as you'll see, several other terms as well. And what Critchley described was a group of sailors who also happened to be former boxers and who presented with the similar profile. And here's 1957. Of great interest, pathological as well as practical, is the fact that this traumatic encephalopathy is a progressive condition. Once established, it not only does not permit of reversibility, but it ordinarily advances steadily. This is the case even though the boxer has retired from the ring and repeated cranial traumata are at an end. Neurologists have visualized this state of chronic traumatic encephalopathy as being based upon multiple minor cerebral contusions, possibly with initial pinpoint hemorrhages later replaced by gliosis, cortical atrophy, and internal hydrocephalus. This was 1957. If someone wrote that right now, it might actually be a pretty good definition and description of what's going on, not only in boxers, but in others who are found to have CTE. But here are the other terms that have been used over the decades for this clinical issue, this clinical presentation that occurs in people with repetitive blows to the head, and then eventually the neuropathological description. But the term that I'm going to be describing more in depth is traumatic encephalopathy syndrome, something that we use now to describe the clinical manifestations of CTE. Football players were actually found to have some of these things way back in the 1930s. It wasn't just boxers, even from the very beginning. And so 1933, in the Homeopathic Medical Society of State Pennsylvania, there was a report of a young football player who did things suggesting the behavior of psychopathic personality, like forging checks, and described the condition we sometimes find in pugilists, boxers, pummeled about the head. In 1937, a former Columbia University football player, Frank Scully, started to interview his former teammates because he just kind of had a sense that there was something going on. And of his just one team, years later, 7 had varying degrees of, quote, dementia, vagrancy, and motor deficiencies. And he believed the conditions seen in boxers could be multiplied many times if applied to football. All right, so let's move ahead. Step 1A for the diagnosis of CT during life is to move beyond punch drunk, because that stuff really was not based on neuropathological diagnosis. It was really just the clinical description, and there may be a very big disconnect. And so now this first step really should be to describe the clinical features associated with neuropathologically confirmed CTE, using especially the most up-to-date neuropathological diagnostic criteria. And so how do we do this? Well, it's based on retrospective reports of the next of kin of deceased individuals. And what we found is that the clinical features that seem to be reported are diverse and nonspecific. So it's going to be a hard job. But let me try to describe what we think we know about the clinical features associated with CTE. First, there are emotional and behavioral control problems, including agitation, rage, short fuse, impulsivity, aggression. but not the type of silliness, social disinhibition, inappropriateness that's seen in behavioral variant frontotemporal dementia. And our group has defined or described this as neurobehavioral dysregulation, this kind of set of emotional and behavioral control problems. And it seems to occur earlier in life, in one's 30s, 40s, 50s. And then there are changes in cognitive functioning, specifically in episodic memory, short-term memory problems. Someone can't make new memories, is rapid forgetting, repeating stories, the same type of thing that we see in Alzheimer's disease memory impairment. There's also executive dysfunction with poor judgment, decision-making, impaired organizational and planning skills, difficulty multitasking, and cognitive disinhibition. In other words, someone acting first and then thinking second. There's also other areas of cognitive functioning that can be impaired, but the two key areas are episodic memory impairment and executive dysfunction. These cognitive changes can often begin later in life, 50s, 60s, 70s. And as the cognitive impairment becomes significant enough to impact daily functioning, to have a true impact on someone's independence, then we would consider that dementia. Dementia from CTE. It's not Alzheimer's disease. It's the dementia caused by the progressive cognitive impairment and functional dependence by CTE pathology. But CTE dementia can easily be misdiagnosed as Alzheimer's disease dementia. We often see this, people who were diagnosed during life as having Alzheimer's disease dementia, when in fact they had no evidence of Alzheimer's disease neuropathologically. Here's a case to illustrate this. This was a 66-year-old former National Football League center. Played 17 years of American football, started playing at age 10, 6 years in the NFL as a center. His family reported 50 concussions, one with a brief loss of consciousness. In his late 20s, he started to have angry outbursts. It wasn't like that before. In his mid-50s, he started to show some mild memory impairment, but he was able to continue to work. But by the age of 64, he retired. He was experiencing pretty significant apathy, just not caring about things, not being interested in things. Also described as having depression and anxiety by his family members. And also in his mid-60s, though, there was a progression of memory impairment. And his outbursts, his angry outbursts, became more common, and then he had problems with navigation, word finding difficulty. He was making phonemic paraphasic errors. He had some gait instability. He was showing masked faces, some mild impairment in instrumental activities of daily living, and he was given a diagnosis of dementia. He died of a heart attack, and his brain was donated to the BU CLF Brain Bank, and Dr. McKee and colleagues diagnosed his brain with stage 4 CTE, severe CTE, and no evidence of Alzheimer's disease or other pathology. But if we saw someone in his mid-60s who would come to clinic with progressive memory impairment, some word finding difficulties, some navigation problems, and perhaps some Parkinsonism, and these neuropsychiatric difficulties, it would be pretty easy to diagnose them with Alzheimer's disease dementia. The thing is that football and other sources of substantial repetitive head impact exposure does not lead to Alzheimer's disease. There's no evidence that playing American football leads to Alzheimer's disease. But for sure, repetitive head impact exposure from football can eventually lead to the dementia from CTE. One way to look at it is through this progression of being asymptomatic or preclinical, maybe at the very beginning of CTE, where there may be these just very small deposits at the depths of the cell side, but very few. And then emotional and behavioral changes, maybe when the disease is progressing a little bit, but possibly when there may be something else going on, like some white matter degeneration. I'll talk a little bit more about that later. And then as it progresses even further, where medial temporal lobe structures are impacted with neurodegeneration of the hippocampus, there can be memory and executive functioning decline. And look at all this atrophy in frontal cortex. And then eventually the entire brain starts being devastated, and the person can go through mild, then moderate, and then severe dementia. And so a question then could be, are there subtypes of clinical features of CTE? I'm not going to spend too much time on this. Early on, we did a paper based only on a relatively small number of neuropathologically confirmed cases of CTE without any other pathology. And the findings suggested that there were indeed two major clinical presentations, one this behavioral or mood variant, and the other a cognitive variant. And that was consistent with other presentations of boxers in the past. And this is just really talking about what I was describing before. And to kind of see it in terms of the differential diagnosis problem, let's just walk through. This is the time when someone might go through this repetitive head impact exposure. And what is going on with this behavioral and mood presentation? Well, it can maybe start during the exposure, but eventually starts to have an impact on someone's functioning, that they're showing this neurobehavioral dysregulation in one's 30s and 40s. After that, we don't have a very clear understanding of the course. When it comes to the cognitive presentation, unlikely any significant change while the person's having this repetitive exposure to the blows. But then when they get to maybe their 40s, definitely 50s, and maybe a more significant decline in 50s and 60s, and then a progressive decline in functioning. But what happens here? If you have someone who presents in their 30s with the kind of problems with control of emotion, problems with impulsive behavior, is that easily diagnosed as early stage CTE? Well, not necessarily. What about the continuation of post-concussion syndrome? Or what about just having some kind of idiopathic depression or non-CTE white matter damage? Or maybe it is related to the tauopathy of CTE, we're just not completely sure. Later on, a differential diagnosis of someone in their 60s and 70s or even 80s who have memory and executive dysfunction and dementia. Well, is it CTE, or can it be a variant of Alzheimer's disease or FTLD or dementia with Lewy bodies when someone has the Parkinsonism? So the differential diagnosis is potentially problematic. And so greater detail needs to be provided above and beyond just this description. And so there needs to be, as step 2 in this process of diagnosing CT during life, the development of diagnostic criteria for these clinical manifestations of CT pathology. There early on were 4 different publications of proposed diagnostic criteria for the clinical presentations of CTE. I'm not going to go into these in great depth, We described this one in 2014. Philip Montenegro was a MD PhD student of mine here who worked with a wonderful group of ours to do this. I'll show that in a second. And then Reams took our TES criteria to try to create a clinical diagnostic criteria, where ours were really just meant to be an initial proposed research-only diagnostic schemata for what we referred to as traumatic encephalopathy syndrome. So this is that 2014 publication. And we used this in our research, and other people used it for several years. And it was then time to do something else with it, to move it forward, to understand, is it working? And if not, what can we do to define in greater detail the diagnostic criteria for traumatic encephalopathy syndrome? And so in conjunction with the National Institute of Neurological Disorders and Stroke, we held a consensus workshop for that purpose. This was in Phoenix back in 2019 with some incredible people who all came together for this process. There were 20 expert panelists from 11 different academic centers representing multiple disciplines, interests, and expertise. We had people from one of our studies called the Diagnosing CTE Research Project that I'll describe in a little bit, as well as outside investigators unrelated to that study. We also had 7 observers, including Walter Koroshetz, the NINDS program director, the director of this institute, and program officials from NINDS, as well as some other key leaders. Here are the people who attended a workshop. I'll give you a second just to look at it. I'm not going to go through every name, but you'll see it's really kind of a who's who of thought leaders in these multiple fields related to the clinical diagnosis of this type of cognitive and neuropsychiatric disorder. And then what we did was we underwent a modified Delphi process to achieve consensus. Our very own Doug Katz led this extensive, almost 2-year process to really achieve consensus amongst those 20 panelists. You can read the details about this in a paper that I'll show you in a second, but the idea was to go through this iterative process of voting and revising to the point where all specific aspects of the criteria would achieve an 80% or more agreement amongst the 20 panelists. The goals originally of that workshop and then to move forward were to develop evidence-informed expert consensus diagnostic criteria for TES, what was viewed as the clinical disorder associated with CTE. The second goal was to improve upon our previously proposed research diagnostic criteria, this 2014 criteria for TES, by maintaining adequate sensitivity, but increasing the specificity for underlying CT pathology, using the defined criteria for CT pathology. And then finally, to develop our own criteria for the clinical presentation, this TES, that would be amenable to future updates without the need for a complete reorganization moving forward as we learned more. At the time of that initial workshop, the review of existing clinical pathological data was based on the information that was published soon thereafter by Jesse Mez and the group here at BU that looked at the ability of those 2014 criteria to actually predict CT pathology. So we used the data that were eventually published in this paper. And as you can see, by using those data from that original TES criteria, we needed to start from scratch again, because those initial criteria were able to rule out CT pathology pretty well, but very little evidence to rule in. In other words, the specificity was horrible. So almost 2 years later of this voting and revising process, we did come up with this publication, the NINDS, Neurological Disorders, NINDS Consensus Diagnostic Criteria for Traumatic Encephalopathy Syndrome. So I'm going to walk through this. I'm going to do it pretty darn quickly and not go through the details for time's sake. You can easily get this paper and look at the specific criteria. Please feel free to contact me or Doug if you have any follow-up questions about it. But it's a stepwise process. Where first you have to establish that the person's had substantial exposure to repetitive head impacts, and then establish if the person meets the criteria for core clinical features, then to assure that it's not fully accounted for by any other disorder. And if all of these criteria are met, then the person would be diagnosed with traumatic encephalopathy syndrome. And then there are criteria to determine the level of functioning. So specific criteria for how independent or dependent the individual is. Each one of these steps has very specific criteria. So for substantial exposure to repetitive head impacts, we go through a whole lot of details. Of course, the primary clinical features, the core clinical features of episodic memory and executive dysfunction, as well as this neurobehavioral dysregulation. It's all laid out. And then not fully accounted for. How do we decide if something is not fully accounted for by other conditions? That's specified as well. And then the level of functional dependence has very specific criteria. So if someone does meet all of those criteria for traumatic encephalopathy syndrome, there's a two-step process to get to some understanding of the level of certainty for CTE pathology. So in other words, first you go through the clinical features, the history of exposure, the level of dependence, all these things. Come up with the TES diagnosis, and then look at supportive features that can help in the kind of prediction of CT pathology, or what we refer to as the level of certainty for underlying CT neuropathology. I won't go into the details of it, but these supportive features include having a delayed course, a variety of motor signs such as Parkinsonism, some psychiatric features, including anxiety, apathy, depression, paranoia. Remembering that these things are not, these psychiatric features are not required for TES, they're just supportive features. But using those things, we can go from having a TES diagnosis to one of these 4 levels of certainty on the bottom for CT pathology: suggestive of CT, possible CT, probable CT, and traumatic encephalopathy with definite CT pathology. For time's sake, I'm going to go through this quickly, but I think you'll be able to understand it if you look at the paper, or if you want a copy of these slides, I'd be glad to share them. But first, you have to meet TES criteria, and if the person is deceased and their brain is examined and found to have CT, well then yes, indeed, they have TES with definite CTE. What if they meet TES criteria, but they don't have the cognitive impairment? They only have the neurobehavioral dysregulation. Well, then they could only be considered to have a suggestive of CT level of certainty. That's because the clinical pathological data really show that cognitive impairment is a much greater predictor of CTE than the neurobehavioral dysregulation. If you do have cognitive impairment, but you are found to have very little exposure to contact or collision sports, then you also could only reach suggestive of CTE. But because all of our data are based upon contact and collision sport athletes whose brains have been examined, and predominantly football players, American football players, these current criteria really are focused on what we know from football players when it comes to gauging the specific amount of exposure to these repetitive blows. And so you can see that if you have a certain amount of exposure, if you then have some of these supportive features, then it would lead to one of these levels of certainty of underlying CTE. I know it looks confusing, but it's actually pretty straightforward. But not as straightforward as the future. Remember, the current criteria just use these supportive features. What we're now moving toward is to have a biomarker that is both sensitive and specific. So someone could meet criteria for probable CTE with biomarker evidence of CTE pathology, similar to what we now have with Alzheimer's disease. And so there are indeed many potential CTE biomarkers. I've been looking at these things for a while, along with other investigators around the country, trying to understand and assess specific biomarkers. And so this was an R01 study of ours that we ran from around 2011 to 2016 called DETECT, or Diagnosing and Evaluating Traumatic Encephalopathy Using Clinical Tests. One of the very, very best acronyms anyone's ever created, in my opinion. So in that study, we did, these are publications from it. I'm not going to go into detail, but it set the tone for what we think we know about potential CTE biomarkers. And so we've done these preliminary studies that have examined non-specific biomarkers like general neurodegeneration and specific biomarkers, meaning perhaps those that measure regional p-tau pathology or the specific p-tau pathology of CTE. I'm not going to go into the details because these are still potential, but there have been several potential fluid biomarkers such as plasma total tau as a measure of overall neurodegeneration in blood. In CSF, we've looked at tau, A-beta, STREM2, a marker of microglial activation. And yeah, we've found some interesting and perhaps important potential biomarkers. We've looked at exosomes in plasma to be able to look at these vesicles that can cross the blood-brain barrier and in which is contained the proteins and other molecules from within the cells. We've done proteomic profiling of these extracellular vesicles in the CSF, as well as in plasma. And now, over the last 5 years, there's been so much growth in new plasma p-tau analytes that look at specific p-tau measures, p-tau 181, 217, and 231 predominantly. These are the ones that have had the most work done in Alzheimer's disease, but perhaps these can be used in CTE as well. And my guess is that if we can use one of these along with some kind of other sensitive plasma biomarker of neurodegeneration or neuroinflammation, we may actually be able to have blood tests that are pretty good at assisting in our in vivo diagnosis of CTE. A very recent study, this was just published, I think, yesterday or the day before by our colleagues at UCSF, looked at plasma p-tau 181 and 217 in 18 individuals who had exposure to repetitive head impacts. It doesn't go into detail about what kind of exposure, but they did meet criteria for TES. What they found in this small sample was that p-tau 181 and 217 were not useful biomarkers of individuals with TES who didn't also have Alzheimer's disease. This was another study of the use of p-tau 181 in retired contact sports. And this study found that although p-tau 181 was associated with abnormalities in corpus callosum and fornix, it didn't predict longitudinal changes in brain atrophy or white matter integrity in individuals who had follow-up evaluations. So those things are pretty preliminary. From our group here, Kate Turk and colleagues looked at cerebrospinal fluid from deceased individuals from the brain bank and found that p-tau 231 in CSF seems to be a promising sensitive biomarker of CTE. And CSF A beta 1 through 42 may also be able to be helpful. So again, we're finding something maybe with a specific p-tau marker, but we're not really sure. I'm going to skip some of these things. Sorry, this is just a repeat of some of the previous slides. So where are we? Well, we have some potential non-specific neuroimaging biomarkers, including things such as structural MRI. We can use diffusion tensor imaging. We can use magnetic resonance spectroscopy. Our colleagues at the Brigham and Women's Hospital, under the overall direction of Dr. Martha Shenton, has been doing some terrific work looking at MR findings in CTE, potential CTE. And Mike Alasko from here has really been taking that stuff a step forward. So I'm not going to go into the details of these things because they really are preliminary, but we've been looking at like limbic system volumes and having a cavum septum pellucidum and cortical thickness, MR spectroscopy, white matter signal abnormalities. What has been really important is to try to look at neuroimaging findings of people whose brains have then been examined after death. And so the group here, led by Michael Alasko, has looked at MRs, magnetic MRI scans that were done during life for clinical purposes, and then looked at how well they related to post-mortem brain findings. That's been incredibly helpful so far, looking at white matter hyperintensities and looking at the volumes and the amount of atrophy in frontal and temporal cortex. So, can we see abnormal CTE in the brain in specific regions, moving beyond MR, moving beyond fluid biomarkers? Can we actually see this abnormal tau species in someone while they're living? And for time's sake, I'm going to let you read all about this on your own, but to basically tell you that we may have a preliminary type of tau radio tracer that could be appropriate for later stage CTE p-tau pathology. So there's a paper of ours that we published a couple of years ago in the New England Journal, where fluortaucipir was used in a bunch of former NFL players compared to some controls. And it did seem to show some promise with differences in superior frontal, medial temporal, a little bit in left parietal, that was pretty strongly associated with the overall exposure to playing football, but wasn't associated with clinical findings. So let me just skip here. I apologize for this kind of speed I have to go through these things. There are still so many questions. Is p-tau the sole culprit for the clinical features? What about those specific early p-tau pathological findings of initial sulcal depth p-tau deposition? Or is it really just the later medial temporal lobe involvement when the hippocampus is destroyed or hurt? Is that what leads to the clinical problems? Or is it just overall neurodegeneration downstream from the p-tau deposition? Well, what about things that aren't related to the p-tau pathology? Inflammatory responses, chronic or progressive changes from a single moderate to severe TBI, or separate white matter damage, either from arteriosclerosis, vascular risks, or perhaps a separate condition where white matter is devastated either from the repetitive head impact exposure or another source. And then there's multiple comorbidities that we have to consider. So there have been several studies now looking at some of these other potential sources of clinical features down the road. So here we have some potential CT biomarkers, but are we there yet? No, we're closer, but we're not yet there. And so we still have some of these important questions to answer. There are several studies that are underway to try to answer it. Colleagues in at Cleveland Clinic in Las Vegas have been doing the professional fighters study. There's the CARE Consortium looking at college athletes, following them longitudinally. Michael Osko and Gil Rabinovich from UCSF have an exciting new study that is doing incredibly well thus far, looking at another PET ligand in former football players. And then the thing that's been taking a huge amount of my time for many, many years, something called the Diagnosing CTE Research Project. And I'll end with just a summary of what we're doing with this. This is not as good as an acronym as the other one, but it is a pretty good acronym. It's a $17 million grant that was originally for 7 years, but because of COVID, we are going to be going an extra year. It should be ending at the end of this November, but we are a year behind. I've had the great honor of working with Jeff Cummings and Eric Ryman and Marty Shenton for these past many years as my co-principal investigators on this, but we also have 50 collaborators across 10 different research institutions, an incredible external advisory board that provides us with guidance, with the goal of this thing being interdisciplinary science, breaking down silos of disciplines, of fields of study, of institutions, with the goals being to collect and analyze neuroimaging and fluid biomarkers for the in vivo diagnosis and detection of CTE, to characterize the clinical presentation, to examine the progression of the course and to refine and validate those diagnostic criteria, and to look at genetic and head impact exposure risk factors for CTE. And then finally, very importantly, to share the data. So we have studied 120 former NFL players across the clinical spectrum, 60 former college players also, and 60 unexposed men, same age. All these people are between the ages of 45 and 74 when they were first seen. They were seen for a 3-day, very extensive evaluation at one of these 4 sites around the country. And you can read all about the details in a paper that Mike Alasko spearheaded that summarizes the entire project and provides a description of the baseline data that came out a year ago. And so this gives you an idea of what we're doing at baseline clinical exams very extensively, CSF, blood, saliva, MRI, diffusion measures, MR spectroscopy, and both amyloid and tau PET. Everyone receives a clinical diagnosis using the traumatic encephalopathy syndrome diagnoses. And initially, everyone was supposed to come back for a 3-year in-person follow-up, but thank goodness we finished everything right before COVID closed everything for baselines. But instead of a 3-year follow-up that was going to be in person, we're now doing 4-year follow-ups remotely. We have this incredible group of people all around the country, including people outside the country, who come together to address all of the incredibly rich database. Stay tuned for some answers for that. I'm not presenting them right now because our first publication is actually coming out very soon. It's been accepted for publication, and we have 2 or 3 more currently under review, and around 25 or more ready to move forward. And then there's a new study that I'm not going to go into detail for time's sake because we need to stop. But this is a way to examine in much greater detail the exposure variables and other variables by studying a large group of 4,800 former athletes, all done remotely through the Brain Health Registry at UCSF, but adding what we're calling a HITS assessment on top of that. So we're studying 1,200 women's former soccer players, 1,200 male former soccer players, and 2,400 former football players. I just want to end with the complexity of it all. Really what it is when we're trying to diagnose people during life is also to try to figure out what is going on. Why would people get it and other people not get CT? And when it does happen, why do some people have a certain type of clinical presentation and others not? This was a model that Alyssa Phelps, who was a research coordinator with us, described, where all of these variables for repetitive head impact exposure, demographics, genetics, lifestyle, medical, psychosocial are involved, as well as the symptoms, the very specific symptoms. And it gets it really complicated. Again, is it just p-tau that results in all that stuff, or are there other things? And so it gets more and more complicated the more we look. And one of the most important things that we're now trying to address is our racial disparities and associated inequities in social determinants of health. That plays a huge role in our current view of this very complex issue. And then there's other psychosocial responses to the retirement from some of these locker room sports that people have been used to having, being part of a team. It's not just you get hit a bunch and you get CT pathology and you get some symptoms. And in fact, it goes way beyond that complexity. This is what I'm going to leave you with. This is using graph theory. I want to just shout out to Diana Tria Rodriguez from our group. She's a PhD student in the graduate program in neuroscience, who's trying to look at this complexity. And you can see these lines connecting some of the key things that we're thinking about here. But it goes beyond that. Each one of those circles has multiple other circles that are associated with it. And it goes beyond that with all of the multiple types of connections. And eventually, we're going to have something like this, where we try to figure out what are the strong and weak connections, how do we bring things together, how are things really related, and try to understand this complexity. And once we can do that, and we can use that to help us with the diagnosis of CT during life, we'll be able to begin clinical trials for that treatment. And if we can detect it early in the disease course prior to symptoms, we can conduct clinical trials for disease-modifying medications, or perhaps to delay or prevent symptoms. And if we can understand the specific risks and resiliency factors for CT, then hopefully we can prevent it. And if not, we'll have to learn from our animal friends. On the bottom it says, I read that story about dementia and former NFL players. Maybe we should reconsider this. So just to end, it's all about this incredible group of people at the BU CTE Center, with whom I've had the great fortune of working, and then well beyond it to all of these folks working at the Diagnosing CTE Research Project, and most importantly, to all of our participants and their study partners and family members. And with that, I'll end 2 minutes late, but thank you very, very much. And I'm going to stop sharing.
Thank you, Dr. Thank you for presenting. Thank you for everyone for joining. I will stick around, so if anyone has any questions for Dr. Stern, please unmute yourself. You can put it in the chat. Again, thank you, Dr. Stern, for this presentation. I found it very insightful.